Just to set the stage for today, as we were thinking in collaboration with the executives and organizers at ARM about today's topic and the panel, we realized that the things we really wanted to talk about were development challenges and solutions. And then we also realized that one of the wonderful things that's happening in our space as acceleration of molecules and products takes place is we're getting to a much more sophisticated understanding of the interplay between a true development challenge and a commercial challenge that affects development. We also realized that we've been graciously given like an hour and a half. Made sense to split this into two panels. So this is the first of a back-to-back set of sessions, which will focus much more on clinical development. And then my colleague John Doyle, who heads up Quintiles IMS's Real-World Evidence and Insight Center, will approach this much more from the commercial development angle. Let me introduce myself. My name is Avi Kulkarni. I'm at Quintiles IMS. We run a stem cell center that is based out of San Diego. So for many of us, it's a short walk from the center to here. We represent all that you would expect from a company called Quintiles IMS, end-to-end services to our biopharmaceutical colleagues in the industry. We have entered the party to some extent late because there's lots of folks who have been pioneering the space, in some case for 2 decades, and the ARM-related activities are themselves more than a decade old. We have ourselves, as a collective institute, Quintiles IMS, been involved in more than 35 clinical studies. We've supported everything from clinical development through to pricing and reimbursement, and some of the insights we hope to draw on today will build off of the work we've done for actual clients in this space. I'm going to ask our panelists to introduce themselves, and we're going to try and make this as thought-provoking a discussion as possible. So hopefully we'll get to the edge where someone is offended. And if not, Adrian will be, my colleague Adrian, who is at the other end of this panel, is going to have to be the one who sweeps up, so to speak, and asks the really crazy questions. So with that, do you want to go next?
Yeah, certainly. So hopefully the microphone's working. Again, good morning to you all. I'm Stuart Albert. I'm the Chief Development Officer for Fate Therapeutics. I have been working in this field for about 10 years now. So basically, I'm still a beginner. Lot still to learn. Fate Therapeutics is a San Diego-based biotech. We work on hematopoietic stem cell-based cell therapies and induced pluripotent stem cell-based therapies, focusing predominantly in immuno-oncology. And our approach to developing those therapies is to basically lock in functional attributes into our cells through small molecule reprogramming and through genetic engineering. And as part of the introduction, Avi asked me to, I think, introduce one challenge. Is that correct?
At least one.
At least one challenge. So I'll introduce my challenge. So I think we've probably all appreciated that cell and gene-based therapies are relatively complex. That tends to make them relatively costly and cumbersome to make. So for an agent that is costly, cumbersome, and complex, it has to be essentially curative to be truly effective or competitive. And so what's the challenge of that? You think that's a sort of interesting point. You've got a curative therapy. Well, if you have a curative therapy, you have the potential to develop it very quickly because your safety and efficacy readouts are going to be coming thick and fast. And therefore, your manufacturing paradigm for your therapy has to develop appropriately quickly. And that's challenging for complex therapies. So I think my basic challenge is, as we get ever better at developing ever more effective therapies, they're going to be developed faster, and that means the manufacturing for those therapies has to keep up with that speed.
Good morning, everyone. My name is Luis Rodriguez Borlado. I'm working at Capricorn. I am VP of Regenerative Therapies. I've been working in this field for almost more than 10 years. Half of my career was done in Europe, and then recently I moved from Spain to LA to work at Capricorn. Capricorn is an LA-based company based on the development of cell therapy. Our product is mainly focused on at the beginning was focused on the cardiac regeneration because the cells that we were using were cardiosphere-derived cells. Recently, we moved to the space of Duchenne muscular dystrophy. And also we have an additional product based also in the cell that are called exosomes. Our trajectory has been quite interesting because we started with autologous cells. We moved from autologous cells to allergenic cells, suggesting that the cells that we are using for these kind of therapies do not stay in the patient for long. And then once that we have that, we move from cells to the exosomes. Still, we think that there is room for both kind of products, for the cells and for the exosomes, probably for different indications and probably for different administration routes. The challenge that we have ahead, I mean, we have a lot of different challenges. I agree with that one in terms of we need to reduce the cost of the manufacturing process. And I think that together with that, something that is going to be very important is to try to establish the bioequivalence between the different products that we are producing. So something that is going to be critical in order to reduce that cost of the manufacturing. And for that, it's going to be also essential to have some kind of bioactivity potency assays that can be in vivo, can be in vitro, that can allow us also to try to establish that bioequivalent between the different batches, between the different donors that we are producing. And for that, it's critical to try to understand the mechanism of action. Because if you don't have a clear mechanism of action, it's going to be very difficult to have a good bioactivity assay that can try to recapitulate the activities that the cells are going to have in vivo. So I think that in terms of preclinical development, this is one of the most important challenges, at least for us. And I think that's it.
That's great. My name is Bob Deans. I'm CTO at Blue Rock Therapeutics. Blue Rock is a very new pluripotent stem cell company that's based in Cambridge, Mass, and has labs both in New York and Toronto. And I think there's a company profile later today by our CEO, Emil Nueser. So I encourage you all to learn a little bit more about us there. We are quite excited because our company has access to a program in Parkinson's disease that's slated to enter the clinic in the second quarter of next year. So we've got a very good program ready to launch and take forward. So my background has been in adult stem cell therapeutics probably for the last 25 years. My triumph is moving out of the adult space and getting into the pluripotent space. I've been at this meeting probably since inception, and I've always been on the adult stem cell side of the game. It's really nice to be in the other chair, another position. So a couple of challenges. I think one for me is more personal. I love Southern California, and I obviously am working on the East Coast. So maybe that forecasts a little bit about Blue Rock's future and where we're looking for additional research centers. I think the interesting challenge for us is to understand how we can learn and use machine learning and AI techniques in mining our clinical data. I think one of the clear things we need is a better understanding of patient stratification that are most responsive to treatment. And I think that by using epigenetic data in early phases of studies, phase 1, phase 2A, we can start to understand the correlation between our clinical surrogates and patients that respond to treatment. So I think we can really boost our learning and capability by understanding what's coming forward in machine learning.
Thank you.
Thanks, Bob. Good morning. I'm Tim Schroeder, CEO and founder of CTI. We are a 18-year-old clinical research organization that specializes in managing cell and gene therapy studies and have about 2 to 3 dozen active programs. And first I wanted to thank Avi and Quintiles IMS for sponsoring the sessions today, obviously very important topics. I think from my side, coming from a solid organ transplant immunology background many years ago, I think the cell therapy reminds me a lot of where we were 20 or 30 years ago in those fields. And the challenge or obstacle that I would like to pose, maybe have some discussion today, is how we handle our clinical development failures. If you think about it there, if you take the CAR-T with Novartis, a monumental success and brings a lot of attention, a lot of investment to the sector, but we will continue to stumble, just like small molecule drug development, biologics, the cell therapy space will have a number of products that do not meet their endpoints and ultimately fail. And I think the challenge is for us as a field is how do we deal with those failures just like we do in other drug development.
Good morning, everybody. My name is Adrian McKinney, again from Quintiles IMS. About 10 years ago, we started a consulting group, an advisory group in Quintiles IMS, and the idea was that over the prior 30 years of drug development and commercialization, a big organization like Quintiles, it must have learned something. There must have been some residual cumulative intelligence and insights. And so our job was to go in and mine them and then bring them out as advice to the industry. I'm now, along with Avi and some other people here, involved with the Stem Cell Center. And again, its purpose is very similar. It's to take all of the emerging knowledge in this very new field with all of these very disparate and various technologies and try and find out what are the sort of commonalities in the way that development can be accelerated. So it can be on the CMC and the manufacturing side. It can be just on pure clinical conduct and getting the right sites. It can be on common approaches to regulatory strategy or evolving approaches and best practices around them to regulatory strategy. But whatever those topics are, and then pricing and reimbursement, to your point, how do you price a cure? There are certain common things that we hope to really focus on in the industry. So I think that two challenges. One challenge for me is that I'm actually a nuclear particle physicist, and the only reason I got into life sciences was about 20 years ago. Somebody confused physicist with physician. And so that's how I ended up here. So that's always a bit of a challenge. The other main challenge, and I don't think it's a downside, it's a lack of capturing upside. I think there's a danger that we don't tease out those collective learnings from across all of the experience in the industry for the greater good. And we'll talk a little bit about some ideas about how to do that.
Thanks, Adrian. Thanks, panel, for the introduction. In terms of format for today, I've put something on screen which just suggests some topics, but I've already accumulated so many questions based on the challenges that the panelists spoke about. We'd like to make sure that we speak through each other to you for another, I'd say, 20-ish minutes, giving a chance to this room to ask us questions also. In the event that we're slow to get a mic and to ask questions, I promise you I have enough here to keep us going all the way to the 45-minute mark. Let me start with the very first question aimed at Stuart, which I think builds off of the development challenges that are related to manufacturing. It's a two-part question, so you feel free to take both or one.
Okay.
So one part is just it felt for a while that as we looked at autologous versus allergenic, it felt as though the solution was going to be not an N of one, but an N of many. So allergenic ought to have triumphed. And yet what we're all speaking about today is Novartis's CAR-T, which is not what we would have, in that sense, expected. And it's overcome lots of challenges to get there. So the question is, what have they done right that we can learn from? So I said there's a two-part question. That's one, right?
Yep.
Here's the second question. It feels as though as we've looked at the complexities around clinical data for proof of efficacy and regulatory acceptance, there's been a lot of movement by the regulators to allow for early enough approval of products with long-tail follow-up registries and so on. But that gets to the clinical evidence package, not necessarily the CMC part of it. So as we think about manufacturing, what modifications would you like our regulators to make that also address the challenge of not having full control over manufacturing, but enough to get you to market?
Okay. Okay, so audience, prepare yourself for a 30-minute monologue while I answer both questions.
Beep, beep, beep.
We can learn the world.
So let me try and address the first question first. So yes, I think if we rewind the clock several years, there was an expectation that expanded allergenic products would win out over patient-specific products. And working in a company that works on both, I'm somewhat unbiased. If we look at potentially why the expectation of allergenic expanded products not winning out hasn't played out to date, or why rather autologous products have been potentially faster to develop. And we'll take the example from Novartis, Kymriah. That program started its commercial development, its industry trajectory, but already having proof of concept in the clinic. So if you think back to 2011 when UPenn did the deal with Novartis and other groups got into this space, there was already clinical proof of concept that a patient-specific genetically modified T cell could be effective. And so we weren't dealing necessarily with the question of could these cell therapies work. The question was really how do we make them work more reproducibly, scale them, etc. So I think, to some extent, the challenge for that particular type of therapy, and use that as an example, but the challenge for that particular therapy was less one of will it work and more of how do we make it work, and therefore the trajectory for development was a little bit more clearer, and that helped hugely for that particular approach. And I think it also helps other approaches that have genetically modified cells as we sort of move forward, because you have an ascribed mechanism of action. To touch briefly on the second point of sort of what can regulators do to help us, or what can the regulations environment do to help us as we move forward. I think, well, I touched on it, and we touched on it. If we're trying to develop therapeutics quickly, we won't always have the opportunity to lock our processes in a traditional manner, such that traditional biopharmaceuticals, the process development is front-end loaded. Large part of the process development is done prior to, let's say, phase 2 studies. There are some changes, but a large part of that still is done fairly early. As we accelerate the development of cell and gene therapies, we have to do large parts of that process development later in the process. If we do that, we run the risk that the process changes that we introduce will introduce changes to the product. And therefore, one of the approaches to trying to avoid that is to have clearly stated goals for demonstrating bioequivalence, demonstrating biocomparability. And I think as we move forward, one of the things that the regulatory environment can help us with is to set clearer expectations as how to demonstrate bioequivalence in later stage clinical studies, knowing that we're going to have to make process changes which may or may not impact the product.
And Stuart, let me interrupt you for a moment and also get Louise involved in this. We talked about assays when you introduced yourself, and bioequivalence touches on that, or probably, in fact, hugely linked right there, right? So if we think about the specific challenges around trying to design an assay when you're not even sure what markers you should be testing for, what do you say to a regulatory agency that says to you, I'm willing to work with you to understand, to let you do the bridging down the road for bioequivalence, but you have to tell me today what you'll be testing for? How do you answer them? What ways can you help that discussion?
I totally agree with the analysis that was done before, and actually I think that is going to be critical to have a bigger window of development of the product, in terms of can be only in phase 2, because so far the knowledge that we have about the mechanism of action that is going to have the cells cannot be tested only in vitro. So we can speculate in vitro, we can speculate even in vivo right now with different mechanisms of action, but the only place in which we are going to have that answer, which bioactivity is good, what bioactivity assay can really tell you what cells are going to be good, needs to be done after having the results of the clinical trials. So far, we can try to explore different bioactivity assays and recapitulate all that information in order to be ready for the first clinical trials in which after obtaining that information, we can try to choose what is going to be the best one, reflected the activity into the patients. So far, we were expecting that maybe we can be able to identify the good cells or the good product based on a very limited number of markers. That's not going to be possible, in my opinion, because there is a lot of heterogeneity between the different donors, especially when you are using product coming from different donors that are going to be relevant in terms of the phenotype of the cells, but maybe not relevant in the activity of the cells. So to try to identify that is not going to be so easy as to have a single marker or a couple of markers. It's going to be much more complex than that. And that's the point, to try to develop that bioactivity assay that can recapitulate all that small difference that we are going to have.
So with apologies to our audience today, it feels almost like a perfect setup to everyone's introduction, because this segues into machine learning, ability to scan for large amounts of data and figure out what's relevant as a signal versus not. So I think that's your cue, Bob.
Yeah, that's very good. Now I'm questioning why I raised that topic. Eh? No, I think it raises a very good point. I think the field has moved from looking at phenotypic or single markers to looking at classes of markers that are defined by pathway and really utilize the ability to probe miRNAs or large data sets that come from transcriptional profiling as a way to characterize the cell product. I think the difficulty there is that many of these assays, it's changing, but many of these assays were not single cell oriented. So you're really looking at properties of the composite population rather than being able to ascribe properties to individual cell types. So I think you've raised a very important point, though, both with bioequivalence testing, that's in line with Stuart's comments about process development and maturing that, particularly in light of accelerated regulatory approval. I think the pressure is there for people to now enter the clinic at phase 1 or phase 2A with the commercially ready process. And in the past, we all knew we had another 3 to 4 years to build our commercial process and bring it forward. Now I think the small biotech company has to be much more proficient in defining their bioprocess, and they have to be ready to use that and employ that in this early accelerated approval framework. So.
And segue from that to someone with a lot of experience in running clinical studies in this space. As you look at the protocols you've been getting at CTI and modifications through the conduct of the study, are you seeing that the things we've talked about, the variance that is necessitated by earlier and earlier push into the clinic, is it causing mid-stage delays as protocols get amended?
Yeah, I think that's a really good point, Avi. If you look at it, probably over the last maybe half a dozen years, we've certainly seen a lot more people get to the clinic quickly, more quickly than in the past, probably with less data and more pressure on them to have that first study be successful. And obviously, every cohort of subjects, you end up with a protocol modification because you're learning as you go. And you don't really have the luxury of saying the point of this study is X, and we'll learn the next set of issues in the next study. We've got to answer as many of those as we can because probably the viability of the product and the company depends on it.
Thank you. Adrian, if I may focus this on something that I know you spend some amount of time thinking about, which is really the clinical development challenge for the system. And it's everything from study, startup, protocol being just part of that through. If you compare, broadly speaking, two sets of experiences, like a Roche-type experience, like large, big pharma experience, versus what we've seen in emerging technologies like Regenerative today. What stands out as the biggest problem that we're seeing at Quintiles IMS, for example, that would truly help, if you could bridge that gap, it would be the sort of the breakthrough moment for us?
I think 2 or 3 things. I mean, one to your point is that protocol amendments are the bane of the industry. It causes the study to stop, time to be lost, patients to perhaps fall out of the study. And there are multiple initiatives across big and small companies to try and address that. Look at the root cause of them. A lot of the time within bigger pharma programs, they weren't set up, the design wasn't thoroughly thought through at the beginning. I think that with these therapies, it is much more emergent. I mean, when we think about supporting a program from a regulatory perspective, you kind of budget maybe for 8 protocol amendments during a year in the early years, just because there's so much flux and change in the understanding. So that's one big thing to address, maybe more cumulative knowledge to try and address the kinds of things that can happen there. I think the other thing is we found getting a patient into a study, a regenerative medicine study, is just a different mindset, it's a different philosophy. The value proposition going in is very different to a small or big molecule in terms of the perceived risk factor, of the level of sophistication of interaction with the cells. I think the second thing is the experience on the trial is very different. It's more binary. You're kind of cured or you're not, and that's a very different patient experience that has to be managed through the clinic. And then finally, the trial's never over. Of course, in other studies for Roche or whatever, there's a long-term cardiovascular outcome side of things, but every study here anticipates maybe a decade of follow-up and maybe sometimes, post-generational follow-ups. And so I think handling those 3 sets of things in a comprehensive way rather than a one-off way every time would be important. And maybe one of the things we're beginning to think is a greater use of patient-reported outcomes is going to be part of that, because all 3 of those things are very much through the lens of the patient, and sometimes that's an easier and less burdensome way of following up than actually future clinical visits all the way through the duration.
Thanks, Adrian. It occurred to me that if I was in a panel like this and I was, let's say, in your seat, or in Tim's seat, and a number of these questions had been coming from, right to left, at some stage you start preparing a set of thoughts, right? This question is going to stay, and this is what I'm going to say about it. and then the topic changes. The moderator terribly refocuses on a different point. And I did that almost 4 times as we moved from right to left, which means that there might have been points the panel wanted to make about questions earlier. So let me stop talking for a moment and just pause and see if things have come up that you wanted to address. Absolutely.
I definitely would. I think a lot of our assay characterization work focuses on the ex vivo properties of the cell. These are living medicines, and I think what we've understood about dose correlations in the CAR T space with response to treatment tell us that we're really not measuring the ability of cells to persist in tissue, the ability of cells to expand in the body, and we don't understand many of the properties that guide migration or tissue localization. So we're not testing for a whole parameter of properties that could make or break whether the cells deliver the ex vivo property that we're measuring. I think we've got to be cognizant that we have a big gap in imaging and the ability to measure these living cell properties.
I think, one other thing occurred to me when it wasn't actually mooted, but the concept began to form that there needed to be some kind of a greater uniformity around understanding of what constitutes a valid assay. Similarly, for all of these post-registrational real-world commitments, maybe there should be some form of more pervasive global registry, such that not every single player in this field has to figure out their own way of looking at the post-treatment effects for an extended period of time. Maybe we need to have more of an industry response for a place where all of that data could be collected in a less burdensome way to any particular company. So, the Framlingham stem cell. Oh, one of my colleagues who deals entirely with orphan designations, he's coined this term, orphanry. So orphanry is because the other thing is, in any of these regulatory pathways, they're all really new. There's lots of nuances about how the arguments are going to be accepted or challenges. And so there's some insider baseball that goes on in actually getting the right designation and positioned in the right kind of way. When it comes to the more operational aspects, sort of the arts of prosecuting the cell in the clinic, we came up with the term celery rather than orphanry. So we might see a whole new discipline called celery about the operations of prosecuting clinical research with cells.
Stuart, it's been a long time since.
Yeah, I guess I might challenge a little bit what you said. And I mean, we do have repositories for information, whether it be during clinical study or afterwards. They're called our national regulatory agencies, and so that information exists. It is centralized. It is compiled. It's reviewed in some depth. I think perhaps the challenge is getting the conclusions from that analysis back out to the user, to the developers, and so we do a good job as an industry of collecting those data.
It's the analysis and the insights.
But it's the analysis and then providing the information back. And in fairness, I think, and this isn't to be sycophantic, but certainly the US FDA do a pretty good job. I mean, they try and give as much advice as they possibly can without breaching confidentiality of different developers and the like. But they generally do quite a good job if you're prepared to listen carefully. And I think the other thing I would say is to touch on that, and it's not to be sycophantic, but we have a question here of sort of regulatory and engaging different geographic requirements. When you think about the complexity and the different sort of the diversity of cell and gene therapy products, regenerative medicine products that we're all developing, it's actually quite remarkable that we have the degree of consensus on regulations that we do, across US, other regions. As a company, we've just sought formal regulatory advice on one of our induced pluripotent stem cell products, and we sought that advice US, Canada, and Europe. I was remarkably surprised on how consistent the feedback was. And that's testament to the fact that we actually do have a regulatory environment that tends to be largely consistent and does share information between the groups. The challenge is then to potentially share more of that information back to the developer.
Yeah. One aspect that I think that is going to be critical is how to integrate all that information, because I agree. I mean, the information is available for everyone, but we need to realize that probably, and I would like to make the distinction between CAR T cells and maybe the regenerative therapy that is probably something completely different, is how we are going to integrate all that information, in the sense that we're not talking about binary product in which they work or they don't work. We're talking about fuzzy set, something like that, something that is diffuse, something that in which the different acids that you are going to give you are going to provide you with different information, and probably is combining all that kind of information in which you are going to have a product that is going to be better or worse. And you can do different kind of tests. And for sure, the different cells are not going to be working always in the same way on all the different tests, but you need to try to establish some kind of matrix in which you are going to integrate all of these. Probably some kind of acids compensate for other ones in order to choose the best product for everyone. And also something that is going to be critical is to understand how to interact that product with every single patient. Because this is, I mean, we have two different players here. We have the product and we have the patients. And the patients sometimes are totally different between them and how they respond to the different kind of products, especially if we are using allergenic cells. We have an important player here that is the immune system. And the immune system is going to be very relevant for the activity of the cells, and the interaction between the allergenic cell that we are working, at least our company and the different patients, is totally different.
Yeah.
And that needs to be answered, especially if you have one administration, but it's more important if you are planning to do more than one administration to the same patient, because that's something that we also need to realize. Is going to be a single treatment, or is going to be multiple treatments with different batch or different banks?
Luis, hold that thought for a second. I want to come back to this. Felt to me that Tim was about to say something a moment back, and then it was Bob right after that.
Yeah. Just two quick comments to Stuart and Adrian's comment. I think in general, organizations like ARM have done a really good job with the regulatory authorities. And if you think about other drug development, what happens over time is precedent drives decision making. And I think we didn't have precedent in cell and gene therapy 5 or 10 years ago. We're starting to see it now. So you can go into the agencies and say to them, in the last program in heart failure, this was an acceptable endpoint, this was an acceptable patient population, et cetera, et cetera. And so I think we need to continue that, but I agree completely with what you said, Stuart, is that there is much more consistency across regulatory authorities in the advice. I think the other thing, Adrian, that you made the point of about the long-term follow-up, one thing that we've been crossed with over the last several years a few times is everybody's thrilled to follow successful patients long-term. Nobody is interested in following the ones where you don't meet your primary endpoint, where the therapy isn't beneficial, where the company goes out of business. And I think there's a whole host of those patients that's only going to get larger and larger, and there's a lot to be learned, as you mentioned, Adrian, from those populations. Sometimes we find advocacy groups are willing to take them on, but it's a difficult group, and clearly, I think back to my original analogy of the solid organ transplantation patients, is there was a registry, a global registry to look at post-transplant malignancy associated with immunosuppression that was funded by multiple companies and was just an independent body. And I think that's something to consider. I think your point's well taken there.
Right.
I was actually taken with some comments that Stuart had made. I'm going to ask you a question. So in looking at CAR T as an example of successful translation, it was clear that Novartis had the pockets that were needed to undertake a very expensive bioprocess and transfer directly from the academic community, bypassing biotech and going into pharma. They de-risked it, so we saw Kite and Juno taking parallel steps. Is this a shift in model where big capital holders are going to look more directly to the academic community for early proof of concept, rather than looking for biotech to go through and serve as a mid-stage development engine? Are we going to see a shift in, and I'm asking you this because you've had both big and small company experience.
Yeah, I mean, I think the simple but not very informative answer is there's space for both. There are always going to be organic, homegrown efforts. There are always going to be, early, mid-stage, late-stage acquisitions. I think that's just part of what we've seen in biopharmaceutical development for decades now. So but with regard to de-risking, and where you have deep pockets and you can de-risk by acquiring assets with clinical proof of concept, then yes, it would make perfect sense. And I think your company is actually developing an academic asset that one would argue is de-risked by virtue of having fetal dopaminergic neuron transplants 20 years ago that happened to work. And so there are approaches here where if one can de-risk, one will. But there's also the speed at which we're developing knowledge means that new opportunities and new therapeutic approaches are coming along every day. So I think there's always going to be, the biotechs, the academics that are coming up with brand new ideas in how to develop, different therapeutics that will be developed in the first instance in those environments.
You think the world has turned, that biotech is starting to look for accelerated approval as a core component of their business model? Or
speak for ourselves,
yeah.
Yes.
Yeah.
Yes. I mean, if you can avoid spending sort of $500 million in development, one will. If you look at the wrapped up cost of, again, going back to earliest CAR T approval, that's still quite an inexpensive therapy to develop. When you put together all of the costs associated with that program, it still looks pretty good.
So yes, that's very good.
So just a time check. We had promised the audience, I had promised the audience, give you a chance to ask questions. If there's mics available or if you, thank you. So all you got to do is raise your hands and a mic will magically appear right here. There's the magic, thank you.
This is a cool mic. I really appreciated that you were discussing kind of the need for better tests of quality of stem cells ex vivo for predicting success in vivo. But one of the things that strikes me as a problem there is that some of the applications where stem cells are very promising and things we look to are things that are very complex etiologies that are difficult to get at even the mechanism of disease progression, let alone the mechanism of treatment. And as soon as we find a mechanism of treatment, it may be that something like stem cells that have very broad actions are no longer the best therapy. So could you talk to that, to kind of the idea that maybe once we understand how they're working, small molecules might be better than stem cells?
Bob, would you like to?
Yeah, big jump.
Thanks.
So I'm not sure that small molecules really can replace cell therapeutics when the modality is really dynamic and responsive to the microenvironment. So clearly, if you've locked in a biologic or drug, you're locked into that modality. The cell population has the ability to expand or not in response to pathology, and they have the ability to modulate, both like modulating early anti-inflammatory effects and then proceeding to look at longer-term, perhaps linked to trophic support of regeneration. So I think there's going to be a very viable and strong space for cell therapeutics that's not replaced by drugs or biologics.
I'm sure the panel agrees, but does anyone have a dissenting opinion? Nope.
No, but the question is really whether in vivo gene therapy will thwart many of the efforts in cell therapy. So
I totally agree with that analysis. At the end, I mean, the kind of response that we are seeing is too complex to be recapitulated by a single molecule, I think. I mean, probably in the future, we're able to identify all the molecules that are playing some kind of activity in that kind of model, and we can try to replace that, but it's going to be a long term until we have able to understand all that kind of complexity.
There's one more question in the extreme, my extreme left. Second row. That is a wonderful mic.
It's different. You're not going to steal that mic.
So I don't want to open a can of worms, but can you comment on potency assays? Because that seems to be the bane of our existence.
Feels like that should be you, Louis, should do that.
Well, yeah, I mean, you need to try to have the potency assay really related with the kind of activity that you expect in the patients. So in our opinion, we are trying to explore different kind of mechanisms of actions. And in that way, we are trying to explore, of course, potency based on the immunomodulation activity of the cells at different levels. Can be T cells, can be macrophages, can be neutrophil cells. Of course, for us, it's important also some kind of regenerative therapy. So we are talking about how that impact on some kind of stem cells that you can establish in vitro in order to see proliferation and differentiation of that. Also, it's relevant in, at least for our cells and for our indications, the cardioprotection, or how the cells prevent the death of different kind of cells after acute insults and that. So some preventing of apoptosis is going to be also very relevant. And finally, is something like, maybe it's not directly related with the mechanism of action, but how is the immunogenicity of the product that you are delivering into the patients? If those cells are going to be rejected really fast, or if they have some kind of immunotolerance after putting that into the patient. So I think that that's 3-4 different potency assays that can try to be established in vitro.
I have a follow-up question to that. Forgive me, sir. Yeah. Well, I was thinking, there's it's a complex issue when your potency is itself an effect. So you're measuring the response to something you've typically injected, and then you say, if I get these response biomarkers, such as immune response, there's a whole range of things you can be checking for. That's potent. And you can define an appropriate level of the marker you're testing for. There's also an element of durability of that response, which gets it to a real sense of complication here. And even if you had a reasonable plateau that you could test for, durability assumes that you're going to be testing for longer than just a minimal period. Because at some stage, the outcome is, it's durable and cured, but what defines that? When do you draw the line to say, if I've seen this kind of a T cell show up for 3 months, that's sufficient. And I think there's a fair amount of onus of proof that we have to generate on this side of the industry to convince the clinicians, the regulators, that the claims we're making around durable potency actually will stay. So I guess the question is, what have we done so far? What are the examples we've seen where we've been able to make those convincing arguments?
I'll tackle that in a second, but I want to throw in one comment. So we can get awfully sophisticated about potency assays, and I agree they're challenging, and they're challenging. They're particularly challenging for gene modified or for cell therapy products that are not modified. As the group, Fate works on modified cells. Part of the reason that we work on modified cells is the modifications that we make to those cells effectively confer potency. So whether it's a small molecule based modification, whether it's a genetic modification, we're modifying to bring about potency, which gives us a measure, at least in the dish or in animal systems, of what that marker of potency consistency is going to be. So that's one approach. But the point I was going to make, which doesn't answer your question, is the patient doesn't care about a potency assay. The patient cares about whether their tumor shrinks, whether their obstructive airway disease is better than it was a month ago. And so we can get awfully sophisticated, and we should, about sort of investigational endpoints and biomarkers of potency. But at the end of the day, the true potency, and back to part of your point, the durability of that potency is how long does the patient feel better for? And that we shouldn't, in all of our sophistication in thinking about the in vitro potency assay, we shouldn't get away from.
So if I may, I'll segue to Adrian, because you talked about patient-reported outcomes and more patient-centric ways of measuring. And I think one way of bridging this is to say it's not just a clinical assay, which is important, but also what are we seeing at the patient response instrument level? What responses are we getting there?
Absolutely. More and more as we're seen many hundreds of small and big molecule clinical programs, a smaller number, perhaps less than 100 of regenerative medicine platforms. And, the question that's coming up more in this new field is, but what does the patient feel? And so it's so much more of an individual outcome that the question, what does the patient feel? Okay. I think that the last thing I might just say is that after looking across all these companies, this field has got so many more snakes and so many more ladders, for anyone familiar with the board game, in between now and the end point, that makes it just absolutely fascinating, and a place where cumulative insights shared across multiple companies will serve us well.
We have a choice. We can either wrap because it's the last minute or two, or we can have one last really focused question. We're going to do a wrap. So thank you. There were a few points that came up today that I think deserve follow-up, right? And I'm not sure we can follow it up here today, so I'll just articulate it. There's a clear need for some degree of a amalgamation of the data that is being generated in this space. You made a point about regulators are giving us access and providing some degree of insight, but it feels as though there's some way of bridging that to the point that was made by Bob about, but we still have to ensure confidentiality and there's protective mechanisms that have to be in place, but we can't have these disparate data sets because this space, more than others, is going to require looking across accumulated data to be able to get the kinds of insights that let us get to market faster and protect the patient as we allow for early and accelerated approvals. So that's one kind of, and there's a question there, how do we do it? Who's going to do it? How is it going to be done? How will it work across multiple jurisdictions? multiple proprietary sets. It just gets very complicated really fast. The second is around, there's a whole range of manufacturing questions that really need to become point-focused, as in, what is the point at which we have got sufficient comfort that we can claim control of the product so that we can get as close to an accelerated approval as possible, and then agree to all the attendant terms and conditions for continuous improvement and testing down the road. Right? The third is around testing. This, in clinical development, unlike our standard methodologies, the ability to develop tests for newer and newer biomarkers that tell us more and more about the product and its efficacy, it's just going to evolve. And we need to be open to this is not closed as much as we need to continue to work on the space, because we'll find out better and better response mechanisms and response measurement mechanisms. There's probably more, so I'll pause. How would the panel like to close this?
For me, something that I learned during this discussion, and I think that is going to be very important, is that, and actually I started this talking about the equivalent between the different products and trying to establish some kind of bioactivity assays in order to explore that. But Stuart mentioned something that for me is going to be very important, and is maybe we don't need to explore all the diversity of the product, but we need to limit the complexity of the product and trying to normalize that. So maybe there is some kind of change in the manufacturing process that can minimize the difference that we observe between the different batches, or even genetic modifications, and maybe that can be a contribution that by CAR T cells on this space in which to, because at the beginning the people was quite afraid of some kind of genetic modification on the cells. But maybe that can be very useful in order to decrease the variability that we have between the different products.
Everyone, a closing comment would be nice, but we've also gotten the warning that we're past our time. So with that, with everyone's permission, thank you for this session. Stuart, Louise, Robert, and Adrian, wonderful session. Thank you.
Thank you.
Thank you.
Please stay for part 2, where I'm going to be in Adrian's unfortunate chair and then get a chance to be grilled by the new moderator. Thank you. I think we can just step out and give it to them. I think the next group should be Mike.